We have also identified proteins that might be important in those processes I mentioned before, which is FMRP. I will mention more about this. The protein is known to bind a specific RNA sequence called G-quartet. This is present in the 3' untranslated region of MMP9 mRNA. There's plenty of this FMRP protein in neurons. This is cell body, this is dendrite, green. So, and some of those green granules contain MMP9 mRNA, shown here with red. So, it is known as FMRP translocates. It carries mRNAs to distant locations. Though interestingly, when we take FMRP knockout, the content of MMP9 mRNA in the synaptoleurosomes does not differ. So, lack of FMRP in those mice could be compensated by some other transporting proteins. Nevertheless, in those synaptoneurosomes, there is more FMP9 activity in this knockout. So, because FMRP has another function, it blocks local translation. So, when MMP9 is missing, the normal I told you that after stimulation, there's increase in this polyribosomal fraction of MMP9 mRNA. In the knockout of FMRP, this is already pretty high, so cannot be further increased. And this is the evidence that indeed protein FMRP may interact in wild-type mice with MMP9 mRNA. When the protein is missing, the antibody against this protein cannot precipitate mRNA and so on. I can't do this. Okay, what MMP9 does, where is there on the synapses? Again, coming to the stools, active, non-active. First of all, if we put the active MMP9 on the top of neuronal culture, we don't see anything. Yes? This is 2 hours following the culture, you see that it looks more or less the same. Yes? Moreover, the intuition tells us that MMP9 is extracellular matrix protease, so should be able to disrupt the extracellular matrix. This is what we also thought some years ago. We don't think this way anymore. So we looked for the major matrix components, such as hyaluronic acid and brevican. And this is with the antibody. This is hyaluronic acid beta protein. This is this culture, treated either with non-active or active protein, no difference. There's no any global alteration in the structure of extracellular matrix. So this enzyme does not produce any major disruption of the structure. But closer look shows that these dendritic spikes, I told you they harbor excitatory synapses. They actually, after treatment with these proteins I mentioned, there is no change in the number of them or density of the dendrites. But when they treated 30 minutes or 90 minutes with the active enzyme, they get longer and thinner. We measure the parameter which is length over the width. Yes? They get longer and thinner. The non-active protein doesn't do anything. This is a measure, so-called cumulative index. Move to the right, says that they are longer and thinner. This is 30 minutes, this is 90 minutes, this is non-active, nothing happens. And this shift over right is actually, so making them longer and thinner, is abolished entirely. This is reflected again by red here. Yes? But this case we use antibody, either to control IgM or specific antibody against integrin beta-1. Integrins are cell surface molecules that allow the proteins to interact, the cells to interact with extracellular matrix. So now when we put the antibody against integrin beta-1 and active enzyme, which is the pink thing, it doesn't happen. So MMP9 really does not disrupt extracellular matrix, but has to reveal a ligand that binds integrin beta-1, which is surface protein translocated, transducing signal into the cells. It is also possible in culture to study plasticity. In this case, the cultures were on a microelectrode array in which we could measure electrical activity of the neurons, hippocampal neurons, grown in culture. And when we were doing this experiment with colleagues from Italy and Germany, actually our experiments in this case were done by Martin Heine in Magdeburg, Katie Conan from Georgetown, from the University of Washington, D.C. She actually, we were doing apparently the same thing, so I showed her results, were published first, that when she treats such cultures with a mixture of compounds that produce some plasticity, especially by activating protein kinase A. This is a major pathway that I will tell you how plasticity looks here. But plasticity has an electrical equivalent. So she just measured the electrical activity by the various means in the sculptures, and denser, darker means there is more of activity here. So before, after this chem-LTP so-called, chemical LTP compound thing, because this increase, sustained increase of electroactivity, she sees the increase, which is actually interestingly inhibited by MMP9 inhibitors, non-specific, MMP inhibitors, non-specific, MMP2/9 inhibitors, and also antibody against interleukin beta 1. What we found that we see in those culture MMP2, but we see also MMP9 less, and MMP9 is the one that is increased in abundance after the treatment, 10 minutes after the treatment. At 40 minutes starts to get back to basal level. The cleavage of beta-dystroglycan is also observed, especially at the 10 minutes. And this cleavage is abolished by GM compound, which is inhibitor of MMP activity. Okay? So there is MMP9 increase, leads to increased electrical activity, is important for this increase, and probably this is MMP9. And morphology, plasticity. What we observe is that this kind of treatment produces increase in the spine size, especially the head width of the spines. Spines may have a shape of a protrusion of a spine, but may have also a shape of a mushroom in which there is a head sitting on the spine. And what we notice, interestingly, is a small percentage, like 10% of small spines only, changes. The big spines do not change, and they get bigger, and they get bigger heads. This is this head, yes, for example, here. And this increase, which we observe in 40 minutes, is again abolished by blocking MMP activity, by the GM compound. Also, those spines that grow, we can detect on them also with this diffuse gelatin live imaging on the confocal, 40 minutes live imaging, also the activity of MMP. So gelatinous activity coincides with those small spines that will grow in terms of having bigger heads. What also happens, this is done in collaboration with Choquet in Bordeaux. On those spines, on those heads, in the postsynaptic density are those glutamate receptors. Yes? I mentioned so far NMD receptor, NR1 subunit, but the most important to transfer the electrical signal through ion fluxes are so-called AMPA receptors. And there's an AMPA receptor called GLU8-1, and this AMPA receptor normally moves around quite a lot on those spines, and after the stimulation, its mobility decreases, this chem-LTP. When we put the inhibitor of MMP, it does not decrease. So one feature is decreased mobility of GLU8-1 receptor. And second feature is, this is this famous super high resolution technology of our Nobel Prize, of which Daniel Choquet is the master as far as the glutamate receptors are concerned. What we notice, what Daniel discovered, that these AMPA receptors go into small groups, which altogether have a diameter, these groups, these clusters of 30 to 50 nanometers, maybe contain about 10 receptor molecules. And what we notice is that those clusters, supercluster, in the bigger groups after CLTP, they are more diffused. You see here this pink signal, AMPA receptor measure live with the specific antibody, fluorescent level. And then after the CLTP, you see less of this diffuse signal on the dendrite, but more of these bigger groups. And then the GM compound again inhibits this effect. And these bigger groups are like this, that are coinciding with the major postsynaptic density protein, which is a synaptic marker, PSD95, marker of excitatory synapses. So, this is what happens in the plasticity. The small spines grow, they have bigger heads. Those bigger heads incorporate more of GLU8-1 receptors that stays immobilized. And this requires MMP activity. You should notice that there is a discrepancy between what I said. That when we put exogenous excess of MMP9, the spine will get thinner and longer. When we put endogenous, they have actually bigger heads, so they are shorter and bigger. We recently think that that we have recently actually resolved this thing by the hypothesis, proving the hypothesis, that it's actually interaction between MMP9 and TIMP that is important. TIMP1 is also released after the CLTP, it's measured here. And we use a trick, again coming to those proteins. This is a measure of MMP9 activity. In blue, the recombinant protein, in a special artificial assay, fluorescent assay. That now TIMP1 is added and blocks this activity, which is reflected here. But now we give an excess of this non-active protein. And non-active MMP9 binds to the TIMP1, scavenges the TIMP1, so it sort of blocks TIMP1 in this case. Yes? So now there's less TIMP1 to block active MMP9. And now we recover the enzymatic activity. So now we have a tool, a novel tool, in which you can mess up with TIMP1. And what happens is, under those conditions, that indeed what we show, that when we block the TIMP1 with non-active, the spines do not grow with heads, but continue to produce these longer and thinner structures, even under chemical LTP, endogenous MMP9. So just to summarize, this is how we believe the MMP9 works at the synapses. Under control conditions, it is produced, released locally from small spines, silent synapses, I don't know, acting through this integrin signaling, produce some signals inside the cells that allow reorganization of cytoskeleton, actin cytoskeleton, growth of a spine into kind of elongated structures, and then within a couple of minutes they are inhibited and produce larger spines, mushroom spines, matured spines, much more of AMPA receptor, immobilized AMPA receptors. And what happens is that now the same signal as before that did not produce an effect, now we produce depolarization. Okay? So this is how we believe MMP9 explains the plasticity, how it's involved. Yes? It is critical in order for the spine to grow, and then more AMPA receptor allow the same glutamate to produce the depolarization, because more of monovalent cations will flow through. Okay, so this was about how we approach learning, memory, plasticity from the C4 MMP9 angle. So this was the good side. Now we have a dark side of the mind, okay, which is mind disorders, or whatever we can start with the mind. So, first with animals, kind of a mind function. We all know that stress affects our mental functions. So this is the study we've done just very recently with Kamil Sandy from Lausanne, in which we look for the protein called nectin-3. Nectin-3 is again postsynaptic protein that interact with nectin-1 that is presynaptic and make kind of a cohesion that could also occur at the synaptic site. And in this case there were rats. Rats were immobilized for 6 hours a day, so they could not move, which is a stressful procedure for them, even in a very gentle treatment, but they don't like it. Either for 1 day or 21 days, it means 3 weeks. And what they noticed, that this nectin-3 level decreases after this 21 days. Where, for example, nectin-1 in the hippocampus does not decrease. We looked for the MMP activity, and using this in situ zymography and gel zymography, we could show that this 3-week stress produces increase in MMP9 activity in the CA1, very specific part of the hippocampus. And this activity was actually specifically MMP9, not the MMP2. And then in the cultured experiments, we could also show that indeed the neuronal treatment with NMDA to produce the NMDA receptor activation, this kind of glutamate receptors, reveals the cleavage product, like in the dystroglycan, in this case nectin-3. And what is the most interesting, this cleavage was entirely abolished by MMP9 inhibitors. So it was evolved by NMDA, general whatever activation of specific NMDA containing structure, synapses maybe, whatever. And then it had to be mediated from MMP9 because the inhibitor of MMP9 blocked it. And then this experiment done in Lausanne, they looked for this mental, what happens with the stress.